Hi Nat5, so we're in the multicellular unit. We're looking at producing new cells, and in particular we're looking at mitosis, so the process of doing that. So what is mitosis? So it's the particular name for saying cell division. It's the posh science name for it. And we use it for different things. If you're a unicellular organism, so a single cell, you literally use this for reproduction, as you just split the cell in two. But for multicellular organisms, we use it for growth and repair. Now, unfortunately, you're going to have to spell this correctly because there is another word that is very similar to this, meiosis, which is to do with making sex cells. And you can see these are really close in terms of what they look like. So you're going to have to spell this correctly because it's one that you cannot be absolutely certain that you got it right if you have something slightly off. So don't put in an extra E, make sure it's mitosis. So here's your stages. So we start off with all your cells' chromosomes that are just muddled up inside the nucleus. They're all mixed together, you can't see them, it just looks like a blob. Now you've even seen that down the microscope when you've stained up cells. So we can't see the chromosomes. But when we start to divide, then something kind of special happens to them. So while you can't even see what's going on, they start to copy themselves. Once they've copied, then they appear. And they appear as condensed chromosomes. So what we have here is two copies. So if this was the original chromosome down here, then what we also have is the copy it made of itself. Now it's really important that they kind of hold on to the copies. So what you have is a central point called the centromere. Now, sorry, there's just an awful lot of words beginning with C, but you're going to have to know them all. So you do need to know the term chromatid. So these are these copies. And you need to know the term centromere, the bit that's holding it together. And while they're like this, the whole thing is called a chromosome. Even though technically what you have is two chromosomes joined together, at this point the whole thing is just a chromosome. Okay, so what you now see is the nuclear membrane disappears and the chromosomes have appeared like this. Okay, now if I tried to draw this out as a human cell, I would have to put 46 chromosomes in there, and that's just way too many. So instead we've just got 6. Okay. So what happens next is that we get some special structures called centrioles, say everything's C's, form at the poles, and our chromosomes line up at the equator. So think of it just like a globe. Okay, so round the middle we've got the equator, and at either end we've got the poles. This is actually 3D, so the center is actually more like a plate rather than a line around the outside, but it doesn't really matter as long as we've got the idea and we use the term equator. So we now have these all lined up nicely. So now what we have is our centrioles and our chromosomes lined up at the equator, and from the centrioles they now shoot out protein fibers called spindle fibers that grab hold of the chromosome. But because they're from either side, effectively what they're grabbing hold of is one chromatid each. Okay. And now they start to pull. So the spindle fibers basically shorten, and as they pull apart, they're going to snap that central bit, the centromere, so it's no longer holding the chromosome together, and our chromatids are going to move towards each pole. Now remember, we originally started off with, so if we originally started off with a chromosome, there's one, it doubled up, okay, to make a chromosome but with two chromatids, and now we've split it apart, it's gone back to one again. Now these ones are identical to the original one. Put the one up at the top. Okay, so what we've done is we've managed to copy and split the exact genetic material inside your cell. This is so important because it means that your cell is going to be able to do the right thing, and that is super important to your body. So finally, we've got our chromatids have now gone up to the poles, and they will now start to do the kind of mushing together that we had at the very beginning. So they will become two new nuclei. New membranes will form around the outside, and we now have two full nuclei inside this cell. That's not going to be there for very long because now the cell starts to divide. Okay, so it starts to pinch off in the middle and becomes finally two daughter cells. Our two daughter cells are now genetically identical to the mother, which is essential because this means that we have exactly the same information in all of them. And you don't want mistakes and differences to start creeping into your cells. The only differences you want are if you have decided that you want a cell to do a particular job, and you've switched on a gene to make that happen. Okay, so one of the ways that you can revise this is to make cue cards, and it's really useful for things where you have a sequence that you need to know. So if you take a sheet of A4 and fold it so you've got 8 equal parts, and then on each one of these parts you're going to write down the stage on one side, and then on the other you're going to draw a diagram. Okay, so here are the 8 kind of statements that I've used in the bits prior. Okay, you can use these exactly, or you could use something similar, but what you're wanting is a description of what's happening at each stage. So if you want to do this, as I say, you can go back and forward, and you can pause this where you need to for it. Still on the cue cards, to make it easier, you might want to use these diagrams. So this is your first 5, 6, 7, and then 8, and then you can label these up if you wish. Put in as much colour. Colour is good, your brain works well on colour, and you can use these, as I say, for many sorting type activities. So you could take your cards, place them diagram up, shuffle them around, you can put them in the correct order. You could do the same with the written sides, okay, as long as you've not put 1, 2, 3, 4, 5, 6, 7, 8 anywhere, which is cheating. You could pick any diagram side, and then you have to say what's on the back, so you can get someone to test you with that for that one. That's quite useful. Or you can have the written one, and then you can describe what the diagram should look like. You can pick up any card and then find the one that follows it and find the one that goes before. All of these are useful if you've got an order that you need to learn. Some key terms that you're going to need to know. Okay, when you see the chromosomes at division, when you can actually, they're out of the nucleus and they're all condensed down, then this is called a karyotype. Now this one that I was using in the diagrams to try and make it a bit easier only has 6 chromosomes, but you should be able to see that what we have are 6 pairs, sorry, 3 pairs to make up the 6. So we've got our 3 different types. Okay, so what we have here is 6 chromosomes. Okay, but what we have is 2 sets of 3. Now if this was a human cell, as I said, that would be a lot more complicated to draw because we have 46 chromosomes, but what we actually have is 2 sets of 23. So there is a name to describe what's going on when you have 2 sets or 1 set. Now we need sometimes to have a 1 set cell. That would be our sex cells, because if you want to have a reproductive system working where you're passing on information, if it's sexual reproduction, you've got 2 parents, you can only give on half of the information from each one. So you have 2 sets in your cell, but you only pass on 1 set in your sex cells, so they are haploid cells. So in this case the haploid number would be 3, but for us it's 23. All of your normal cells are diploid because they have 2 sets, and that's your terminology. So the most important thing here is that we maintain something called the chromosome complement. So what that means is that the diploid mother cell produces 2 identical diploid daughter cells. So you start off with 2 sets and you end up with 2 sets. Okay, no matter what. So if you started off with, say, 46 chromosomes, then it divided, you would end up with 46 chromosomes. It doesn't matter if it's 1 division or 10 divisions. If they ask you how many chromosomes have you got in the daughter cells, it's always going to be the same as the mother cell. It's not doubling every time, and the only time that it would half is if you were going to a sex cell. If it's not maintained, I hope you can see that that's just not going to be good for the cell. It could mean the cell just can't function the way it wants to, and it should, or it could be something more drastic. It could be doing something it shouldn't, and that can either be that it's not working well or that it is far too selfish, and effectively that could be cancer. Okay, that's us.